Welcome to our deep dive into gene expression analysis, a powerful tool that helps scientists understand how genes are turned on or off in different cells. By studying gene expression, researchers can uncover the mysteries of how our bodies function, respond to diseases, and adapt to various environments. This process involves measuring the activity of thousands of genes at once, providing a snapshot of cellular activity. In this video, we'll explore the basics of gene expression, the methods used to analyze it, and why it's so important in modern biology and medicine. Let's begin our journey by understanding what gene expression really means. Gene expression is the process by which information from a gene is used to create a functional product, like a protein. Not all genes are active at all times, some are switched on or off depending on the cell's needs. This selective activation allows cells to perform specialized functions. For example, muscle cells express genes that help them contract, while nerve cells express genes involved in transmitting signals. Understanding which genes are expressed and to what extent gives us insight into cellular behavior and health. Analyzing gene expression helps us answer critical questions in biology and medicine. For instance, by comparing gene expression in healthy and diseased tissues, scientists can identify genes involved in illnesses like cancer. This knowledge can lead to new diagnostic tools and targeted therapies. Additionally, gene expression analysis is used in agriculture to develop crops with desirable traits, and in environmental studies to monitor how organisms respond to changes. Applications are vast and impactful. The heart of gene expression is the central dogma of molecular biology. DNA makes RNA and RNA makes protein. This flow of information is fundamental to life. DNA, our genetic blueprint, is transcribed into messenger RNA, mRNA, which is then translated into proteins that carry out cellular functions. By measuring mRNA levels, scientists can infer which genes are active and how much protein might be produced. This concept is key to understanding gene expression analysis. There are several methods for analyzing gene expression, each with its own strengths. Traditional techniques like northern blotting have given way to more advanced methods such as microarrays and RNA sequencing, RNA-seq. These technologies allow researchers to measure the expression of thousands of genes simultaneously, providing a comprehensive view of cellular activity. Choosing the right method depends on the research question, available resources, and desired level of detail. Microarrays are one of the earliest high-throughput methods for gene expression analysis. In this technique, thousands of DNA probes are attached to a solid surface. When labeled mRNA from a sample is added, it binds to complementary probes, revealing which genes are active. Microarrays are cost-effective and allow for the simultaneous analysis of many genes, making them a popular choice for large-scale studies. RNA sequencing, or RNA-seq, is a cutting-edge method that provides a detailed and accurate picture of gene expression. Unlike microarrays, RNA-seq doesn't rely on pre-designed probes. Instead, it sequences all the RNA in a sample, allowing for the discovery of new genes and rare transcripts. RNA-seq has revolutionized the field by offering greater sensitivity and the ability to detect subtle changes in gene expression. For analyzing gene expression, samples must be carefully prepared. This involves isolating RNA from cells or tissues, ensuring it's pure and intact. The quality of RNA is crucial, as degraded samples can lead to inaccurate results. Researchers use specialized kits and protocols to extract and assess RNA, setting the stage for reliable gene expression analysis. Once RNA is isolated, it's often converted into complementary DNA, cDNA, through a process called reverse transcription. This step is necessary because DNA is more stable and easier to work with than RNA. The resulting cDNA serves as the template for further analysis, whether using microarrays or sequencing technologies. In microarray experiments, cDNA samples are labeled with fluorescent dyes. These labeled samples are then hybridized to the microarray, where they bind to specific DNA probes. The intensity of the fluorescence at each spot indicates the level of gene expression. This step is critical for generating readable and interpretable data from microarray analysis. For RNA-seq, the cDNA is sequenced using high-throughput sequencing machines. These instruments read millions of short sequences or reads from the sample. The resulting data is massive, requiring powerful computers to store and process. This step generates a comprehensive dataset that reflects the abundance of each RNA molecule in the sample. Raw gene expression data can be influenced by technical variations, such as differences in sample preparation or sequencing depth. To ensure accurate comparisons, scientists normalize the data. Normalization adjusts for these variations, making it possible to compare gene expression levels across different samples and experiments. This step is essential for reliable analysis. Before diving into analysis, researchers perform quality control checks on the data. This involves looking for outliers, checking for consistent results across replicates, and ensuring that the data meets certain standards. Quality control helps catch errors early, preventing misleading conclusions and ensuring the integrity of the study. One of the main goals in gene expression studies is to identify genes that are expressed differently between conditions, such as healthy versus diseased tissues. Differential expression analysis uses statistical methods to pinpoint these genes. The results can reveal key players in disease processes or responses to treatments, guiding further research and potential therapies. To make sense of complex gene expression data, scientists use visualization tools like heat maps and volcano plots. These graphical representations help highlight patterns, such as groups of genes that are co-expressed or significantly changed. Visualization makes it easier to communicate findings and spot trends that might otherwise go unnoticed. After identifying differentially expressed genes, researchers often perform functional enrichment analysis. This technique determines whether certain biological pathways or functions are overrepresented among the genes of interest. It helps connect gene expression changes to broader biological processes, providing deeper insight into the underlying mechanisms. Gene ontology (GO) analysis is a common approach in gene expression studies. GO provides a structured vocabulary for describing gene functions, biological processes, and cellular components. By mapping genes to GO terms, scientists can identify which functions are most affected in their experiments, offering a clearer picture of the biological impact. Pathway analysis goes a step further by examining how groups of genes interact within biological pathways. This approach helps researchers understand how changes in gene expression affect entire systems, such as metabolism or immune response. Pathway analysis can reveal potential targets for drug development or highlight key regulatory networks. Recent advances have made it possible to analyze gene expression at the single-cell level. Single-cell RNA-seq allows scientists to study the diversity of cells within a tissue, uncovering rare cell types and subtle differences. This level of detail is transforming our understanding of development, disease, and cellular complexity. Traditional gene expression studies analyze bulk samples, averaging signals from many cells. In contrast, single-cell analysis examines each cell individually. While bulk analysis is useful for general trends, single-cell approaches reveal the unique characteristics of individual cells, providing a more nuanced view of biology. Gene expression analysis has revolutionized medicine. It's used to diagnose diseases, predict patient outcomes, and personalize treatments. For example, cancer subtypes can be identified based on gene expression profiles, guiding therapy choices. This approach is paving the way for precision medicine, where treatments are tailored to each individual's genetic makeup. In cancer research, gene expression analysis helps identify genes that drive tumor growth or resistance to treatment. By comparing tumor and normal tissues, scientists can discover biomarkers for early detection and targets for new drugs. This knowledge is critical for developing more effective and less toxic cancer therapies. Gene expression analysis is also vital in studying infectious diseases. By examining how host and pathogen genes are expressed during infection, researchers can uncover mechanisms of disease and immune response. This information guides vaccine development and helps identify new strategies to combat infections. Gene expression patterns change dramatically during development. By analyzing these changes, scientists can map how cells differentiate and tissues form. This knowledge is essential for understanding birth defects, regenerative medicine, and the basic principles of life. In agriculture, gene expression analysis helps breed crops with improved traits such as drought resistance or higher yields. By understanding how genes respond to environmental stress, scientists can develop plants that are better suited to changing climates and growing conditions. Gene expression analysis is a powerful tool for monitoring environmental health. By studying how organisms respond at the genetic level to pollutants or climate change, researchers can assess ecosystem health and predict future impacts. This approach supports conservation efforts and informed policy decisions. Despite its power, gene expression analysis comes with challenges. Technical variability, data complexity, and the need for robust statistical methods can complicate interpretation. Researchers must carefully design experiments and use appropriate controls to ensure reliable results. Ongoing advances in technology and bioinformatics are helping to address these challenges. Bioinformatics plays a crucial role in gene expression analysis. Specialized software and algorithms are used to process, analyze, and interpret large datasets. Bioinformaticians develop tools for normalization, statistical testing, and visualization, making sense of complex data and enabling new discoveries. Machine learning is increasingly used to analyze gene expression data. These algorithms can identify patterns, classify samples, and predict outcomes based on complex datasets. Machine learning enhances the power of gene expression studies, enabling more accurate diagnoses and personalized medicine. Gene expression analysis is often combined with other omics data such as genomics, proteomics, and metabolomics. Integrative approaches provide a more comprehensive view of biological systems, revealing how genes, proteins, and metabolites interact. This holistic perspective is key to understanding complex traits and diseases. Many gene expression datasets are publicly available in databases like GEO and ArrayExpress. These resources allow researchers to access and reanalyze data from previous studies, fostering collaboration and accelerating discovery. Public databases are invaluable for validating findings and exploring new questions. Gene expression studies, especially those involving human samples, raise important ethical considerations. Issues like informed consent, data privacy, and responsible data sharing must be addressed. Ethical guidelines ensure that research respects participants' rights and promotes trust in science. The field of gene expression analysis is rapidly evolving. Advances in technology such as long-read sequencing and spatial transcriptomics promise even greater resolution and insight. As methods improve, we can expect more precise, comprehensive, and impactful studies that will shape the future of biology and medicine. Spatial transcriptomics is an emerging technique that maps gene expression within the context of tissue architecture. This approach reveals not only which genes are active, but also where they are expressed, providing a new dimension to our understanding of biology. Spatial data is crucial for studying complex tissues and disease microenvironments. Epigenetic modifications, such as DNA methylation and histone modification, play a key role in regulating gene expression. These changes do not alter the DNA sequence but can turn genes on or off. Studying epigenetics alongside gene expression provides a more complete picture of gene regulation and cellular identity. Not all RNA molecules code for proteins. Non-coding RNAs, such as microRNAs and long non-coding RNAs, play important roles in regulating gene expression. These molecules can enhance or suppress the activity of specific genes, adding another layer of complexity to gene regulation. Gene expression patterns can change over evolutionary time, leading to differences between species. By comparing gene expression across organisms, scientists can uncover the genetic basis of adaptation and evolution. This research sheds light on how new traits arise and how species diverge. In neuroscience, gene expression analysis helps unravel the molecular basis of brain function and disease. By studying which genes are active in different brain regions or cell types, researchers can link gene activity to behavior, cognition, and neurological disorders. Gene expression analysis is crucial in immunology, where it helps map immune cell responses to infections, vaccines, and autoimmune diseases. By profiling gene activity in immune cells, scientists can identify key regulators and potential therapeutic targets for improving immune health. Pharmacologists use gene expression analysis to understand how drugs affect cells and tissues. By monitoring changes in gene activity after treatment, researchers can assess drug efficacy, identify side effects, and discover new therapeutic targets. This approach supports safer and more effective drug development. In plant science, gene expression analysis reveals how plants respond to environmental stresses, pathogens, and developmental cues. This knowledge is used to breed more resilient crops and understand fundamental processes like flowering and fruit development. Microbiologists use gene expression analysis to study how bacteria and other microbes adapt to their environments. By tracking gene activity, researchers can understand microbial metabolism, resistance mechanisms, and interactions with hosts or other microbes. Biotechnology companies harness gene expression analysis to engineer organisms with desirable traits, produce biopharmaceuticals, and optimize industrial processes. This technology is at the heart of innovations in synthetic biology and bioengineering. While gene expression analysis is powerful, it has limitations. Technical artifacts, sample quality, and interpretation challenges can affect results. It's important to validate findings with complementary methods and remain cautious about overinterpreting data. Awareness of these pitfalls ensures more robust and reliable research. Gene expression analysis has transformed our understanding of biology, medicine, and the environment. By measuring which genes are active under different conditions, scientists can uncover the molecular basis of health and disease, drive innovation, and improve lives. As technology advances, gene expression studies will continue to open new frontiers and answer fundamental questions about life.